All right, Miss O'Leary here, back again to talk about some PCR. So the first step in PCR is basically deciding what you want to amplify or what you want to make those billions of copies of. Once you do that, and you've identified what is labeled here as your region to amplify, you have to select your primers. And this is where the 5' and 3' are really important. Um, so one of the things you should be able to do is look at the DNA templates and then remembering, which we'll talk a little bit more about later, that the primer is also DNA and has to attach to the template. Can you label the 5' and 3' ends of the primer? Now, one of the things also to remember is that DNA can only be extended on a 3' end. So that means that the 3' ends of the primers must both be facing the region to amplify. So just one more time, the primers have to be facing with the 3' ends in towards the region that you're amplifying. And then once you do that, you do have to know a little bit about your region that you're amplifying in terms of sequence so that you make sure that your primers are in the right spots. These primers, basically, if I know what the sequence is on my DNA template, I can log on to a company website and say, I want a primer with this sequence running from 5' to 3', and they will synthesize it for us. And you can see the outcome, the amplicons, or basically the copies generated by PCR using primer 1 and 2. You should get double-stranded pieces of your DNA of just that amplified region. Okay, so the main parts of PCR revolve around the primers, basically isolating the region that you're interested in, having a polymerase that can actually build new strands of DNA, and then having free nucleotides. That's DNTP, those are the nucleotides. And so with those things together, if you run them through a different series of temperature changes, that will allow them to all come together to make new copies of that region of DNA. So the temperature cycling, you first have to heat up the DNA so that the strands break apart. It's also called denaturing, specifically breaking the hydrogen bonds. It has to be cooled down so that the primers are able to rebind or bind to the DNA. And then you heat it up a little bit, but not very much, and allow the DNA to be replicated. Now, one of the things that's important about this is that this cycle happens usually around 30 times. And because of the temperature changes, we have to be careful about what enzymes we use. So this is a DNA polymerase that makes copies of DNA. It's an enzyme, and as we know about enzymes, if they get too hot, this should be in reference to your lab from the last unit, they will denature. So we have to have a polymerase that will hold up to 94 degrees Celsius. Okay, that's very warm. So kind of like the hot block that you used in class, very, very warm. So the polymerase that we use is Taq polymerase, TAQ, and it was originally isolated from bacteria that lived in thermal vents. So because it was in thermal vents, those bacteria, their enzymes had to be able to withstand the high temperatures. And so by isolating that, we were able to use it in the lab. So this is a picture of what you get after each of the cycles. So notice that we've drawn out the first 3 cycles. You only get those amplified regions after the 3rd cycle. So you have to go through a couple of cycles before you're actually going back and forth between the 2 primers to get those millions of copies. I highly recommend going through this tutorial. Go through the amplification step by step. It will show you how the primers bind, what the amplification looks like, including the kind of differences in the first 3 steps versus those after. And then the graph will give you an idea of how we go from just a little bit of DNA to these millions of copies. This video is also useful, so if you want to go through kind of the step-by-step big idea of PCR, Amoeba Sisters are always a good idea, and they're cute to watch. Okay, so as you are reviewing the process of PCR, these are the kinds of things that you should be able to explain. Can you talk about the outcome of PCR? So what do we get out of it? And why is that important? How do we pick the primer? So what do we have to know about sequence. What do we have to know about the 5' and 3' directions? What is the purpose of the temperature cycling? Why do we have to use Taq polymerase instead of polymerase, for example, isolated from human cells? How does the process actually isolate a specific area to copy versus a whole chromosome? So this you can usually figure out by trying to draw through the first 3 stages. That last tutorial would be helpful for that. And then how does all of this relate to the structure and directionality of DNA? So the second part of our PCR lab is the electrophoresis. Um, so this video is really helpful, but just to go through a few of the main points on the right here, you have um, electrophoresis gel, which is kind of like really hard jello, if you've had jello or know what jello looks like. Um, an important aspect of DNA is because of the phosphates. If you go back and look at the phosphate groups, the phosphates are negative, which means overall our DNA has a negative charge. And what we know is that opposites attract. So if you put your DNA in the negative end of this gel and run electricity through it, it will move and be attracted towards the positive end. And so the DNA will move down this gel with the help of electricity. Now, whenever we stop the gel, the pieces that were smaller will move faster towards the positive end because it's easier for them to get through the gel, and the longer pieces will be closer to the wells. I always think of this kind of like a jungle gym. So if you have a really complicated jungle gym, a 3-year-old is going to have an easier time getting through it than a 15-year-old because they're smaller. They can get through those little crevices and things a little bit faster. So same sort of thing here. Small pieces move faster. And you get something like this at the end. So we would go through the electrophoresis and then the visualization of it, which there's more about that in your lab. So you can see on the left, though, we have a marker, or this is also called a DNA ladder. So this is, again, something that we would just buy online. We would say we want a 100 base pair ladder. And what it comes with is DNA of all the different sizes from 100 going up by hundreds to 1,000. And so when we run this on our gel, that gives us a reference point since we know that each of these bands is 100 base pairs different. So we start at 100, 200, 300, 400, and up to 1,000. So that means if we look at our samples, if we pick one of the bands, for example, this third band down in sample A, if you follow over to the left, if we would draw a line from that, we can see that it is between 600 and 700 in the marker. So that tells me that the DNA fragment from this band is between 600 and 700 base pairs. Now that in itself doesn't tell me anything, but when I have more information or something to compare it to, that is helpful. And that is how you will interpret your gels from our lab. This is useful just to see what electrophoresis can be used for in terms of profiling and fingerprinting. So if you ever watch the CSI shows, they do a little bit of this. They make it look a lot easier and faster than it really is, but the process is pretty similar. So you can basically compare um, potential suspects, if we're talking about like a crime scene, to a known sample or something that would be found somewhere. So there's different ways to do that. So take a look at these; they'll be interesting for you. Okay, so what about bacterial DNA? Um, so we in the DNA fingerprinting, we're talking about looking at samples from people. In bacteria, you would have basically your samples are going to be different types of bacteria instead of people. And then think about how this relates to the selection of primers in a eukaryote when we're trying to isolate a gene from humans versus trying to figure out different types of bacteria. So that's something that you will be examining in the lab. Okay, so how does PCR relate to DNA synthesis? So just to review, we have this idea of making copies of chromosomes, and that is important for cell division. So our DNA basically inherently, through evolution and gain of these different molecular functions, is able to replicate DNA in the cells. Um, One of the things that we have to reference is also what does the DNA look like um, in different times in the cell. So you can see, I mentioned this in the previous video, supercoiling. So the bottom right shows you eukaryotic chromosome and those histones, octamers that I mentioned. So basically, how does it condense into those shapes we recognize, kind of like the condensed chromosomes? But the same thing is also true of the circular DNA in bacteria. So the chromosome is compacted about 1,000-fold to fit within the cells. The reason that this is important is because when you replicate the DNA, even though it may not always have this chromosomal structure, it is always condensed, which means that you can't just denature the whole chromosome like you can for PCR. You can't denature the whole thing. You can only open up a little bit at a time because it is very tightly condensed. So that means we have to start small. And to that end, we have something called the origin of replication. And this gives us what we call the replication bubble. So on the left over here, if you look at it, the ORI, the little pink, represents the origin of replication. So you can see that there's a space here where the DNA is creating this bubble shape. And then what happens is this bubble basically gives you single-stranded DNA on both sides. And then both sides will get new DNA replicated onto it. And then as it goes around, this bubble expands. Okay. Eventually, meaning that you get those two circles that are linked together. A very similar thing in a eukaryote. In a prokaryote, you only need one origin of replication because they're pretty small circular chromosomes, so it goes pretty fast. In a eukaryote, you have lots of origins across the chromosome. But the same basic thing applies. A little bubble is opened up, you get new DNA on both sides of the bubble, and then eventually, as these bubbles move away from the origin, they will run into each other. So you get, instead of just these pockets of duplicated DNA, eventually you'll get the entire chromosome duplicated. One of the first steps here is basically to create the bubble. So you can see on the right, we have these things called single-stranded binding proteins and helicase. Helicase is only present in replication. Sometimes people get it confused for something you would see in transcription, but it's only replication. So helicase is an enzyme that's made of multiple parts that basically goes through and busts apart the hydrogen bonds in the DNA. The single-stranded binding proteins stabilize the DNA. So if you imagine if these pink little circles were not here, all this part that was open behind the helicase would just snap back together because the hydrogen bonds would naturally reform. So it's important to kind of keep them stabilized as single strands until the DNA can be replicated. The primers are also needed in replication, just like we talked about in PCR, except in this case, instead of being made of DNA, they are made of RNA. So this is for a number of different reasons. The biggest picture one being that's how the process evolved, but also RNA is more stable, single-stranded. Your teacher will likely talk to you about the difference between RNA and DNA, but it's very similar. It's ribonucleic acid instead of deoxyribo. And so that means it has another oxygen on it. But that extra oxygen actually stabilizes it. So it's a little easier to plunk down as a single-stranded piece. And then we have an enzyme. The enzyme primase is actually the thing. I always think of this as kind of like a stapler. Primase staples in these pieces of primer. And then just like in PCR, we do have a polymerase that can extend off of these primers to make DNA strands. The difference being, because the primers are RNA, they cannot be part of the end product. So there has to be a step at the end that they are removed. Excuse me. So this shows a little bit about what the replication bubble looks like. So if you start in the center at the origin of replication and put a primer on both sides of your bubble, what you will find is that the primer on the bottom can only be extended on the right. The primer on the top can only be extended on the left. And where those easy extensions happen, those are the leading parts or the leading strands on each side of the bubble. On the other parts of it, we still have to replicate in the same direction. So basically, if I take this right half, the bubble has to keep opening away from the origin to the right. But I somehow have to replicate to the left. So what happens is that you get a new primer. So there's a primer that's a little bit to the right of the first one, but now there's a free 3' end on it that can be extended until it runs into the last primer. And then we get another primer a little bit closer to the fork being here. And then it gets extended until it runs into the last one. And that happens on the other two parts of the bubble. And those are called the lagging strands. If you've heard the term Okazaki fragments, that's what you get here. Or if you see this come up, Okazaki fragments, these are these little pieces. But just remember that the overall direction of replication has to stay the same. So that's why you have a leading and lagging strand on each half of the bubble. You can see just one side of the fork here. Um, the fork, that would be like the left part of the bubble that's opening here. Because it doesn't replicate the same way in all the parts, we call this semi-discontinuous replication. So continuous would be where the leading strands are, and discontinuous is where the lagging strands are. So semi-discontinuous means it's partially not continuous. And we also call it semi-conservative replication because the outside original strands of DNA are not changed at all. They are conserved. And so that means that half of each of the new replicated pieces of DNA are conserved. And so we call that semi-conservative replication. This involves quite a few enzymes here. There are some good videos too that will help you to see this. We mentioned helicase before, that goes through and busts apart the hydrogen bonds. Gyrase is actually also pretty important. It makes little snips in the DNA so that the twists unwind. Basically, think about if you have like a knot in a necklace or a string and you just pulled it tighter, it would just get worse and worse. Like the tangling would get worse. But in this case, gyrase basically relieves the tension so that you don't get tangles in the helix down the way. The single-stranded binding proteins are seen out here. You can see primase. Primase does work on both strands, but you can kind of see this one doing the lagging strand down here. And then DNA polymerase 3 is the enzyme that comes next. So these red donut-looking things are DNA polymerase 3. They're basically the equivalent to the Taq polymerase in PCR, and they make the new DNA strands. And so that's up to where we already know. But if you remember, I mentioned that RNA and DNA cannot stay in a strand together. So these green pieces of RNA have to be replaced. And so DNA polymerase 1 removes the primers and replaces it with DNA. So you basically take out where that little staple was and replace it with a strand. However, because it's not a continuous replication there, you get these little gaps in the backbone that you can see here with this yellow enzyme. And the last enzyme that we have is DNA ligase. And what it does is it fills the gaps in the backbone and makes bonds here. So it basically joins the fragments together. So think ligase, ligates, and ligate means to put together. So I would definitely go through these different videos and watch this as well. It's a lot easier to see in action than to just listen to me talk through it. So that one in particular is pretty good with the Amoeba sisters. And then the next question you want to look at is, how do PCR and replication compare? So go back through these different pieces of information that I just mentioned about replication, and how does it compare to what we talked about with PCR? Okay, now that was what we considered the DNA replication model. A model is something that you can use to understand and predict. The realistic view of what replication looks like is a little bit more complicated. So we showed all of the enzymes moving independently on the leading and lagging strand. But in reality, all of the enzymes, like you can see in this picture, are held together in one big complex. So instead of having that nice little bubble with everything moving independently away from the center, it's a little bit more complicated because the lagging strand actually loops out. We actually call this tromboning, if you've ever seen someone play a trombone. So it loops out and then is brought back together, loops out and is brought back together. It's much harder to draw, but if you kind of have the understanding that even though we're drawing them in the model kind of independently, Because it always progresses in the same direction, we have this big complex happening. And you can see that in real time in this video, which I'll just play a little bit of here. So you can see that there is still a leading and lagging segment, but they're just happening at kind of different rates using this whole enzyme complex. Okay, so that's where I'm going to stop for this particular video on PCR and replication. I will do another one in a little bit related to how all of this gives rise to mutations that will connect us back to some of what we talked about with evolution.